And now we will move to our next speaker, Philip Shaman. Philip is doing his PhD under the supervision of Tiago Branco at the Sainsbury Wellcome Center for Neural Circuits and Behavior in University College London. He will talk about his recent bioprint, which will be posted in the chat shortly. They investigated the navigation strategies that mice use while seeking shelter in the new environment. His talk is titled Mice Learn Multi-Step Routes by Memorizing Subgoal Locations. Stage is yours.
Thank you. All right, thanks for joining me. So in this brief talk, I'll be introducing our recent preprint, which examines how mice navigate to safety when multiple step routes are required. First, I'll introduce the most common framework for understanding navigational strategies, which is basically lifted from the hippocampus as a cognitive map by O'Keefe and Nadell. And in this framework, on the one hand, you have what's called taxon navigation, which relies on proximal cues like a visual beacon or an odor trail. Learning occurs gradually by practicing movements or routes over and over. And so an animal could learn a multi-step route by chaining together a series of learned actions with respect to local cues, like a set of trees. On the other hand, you have cognitive map strategy. This strategy relies on a sense of place. Its learning occurs in an all or none fashion based on curiosity-driven observation of how the environment is structured. And so a multi-step route could be computed by, say, simulating paths within an internal representation of the environment and then selecting the best ones. However, I'd like to also note that in this dichotomy, it's based largely on experimental studies with very particular mazes and trial structures, or else in simple open field environments. And so it's uncertain how this maps onto more naturalistic scenarios. And our lab has found escape behavior in particular to be a useful complement to more traditional methods for studying navigation for a few reasons. For one, we can reliably trigger goal-directed routes to safety using innately threatening stimuli, and this allows for robust analysis of the animal's paths. Previous work also indicates that the same toolbox of spatial reasoning that animals use for foraging is also engaged during defensive behavior. And this is instinctively motivated behavior, which reduces bias in that we don't need to select particular training protocols, and it allows us to look at spontaneous strategies that mice use when exploring novel environments. And so here's what our main setup looks like. We have an elevated platform with a mouse, a plastic box, which it finds to be sheltering, an obstacle in the center of the platform, and a threat zone, which at certain points when the mouse enters this zone, will trigger an auditory threat and the mouse will find its way back to the shelter. So again, it's entering this arena for the first time in its life. It explores for about 10 minutes. We trigger threat trials when it enters the threat zone. It leaves the shelter and we do it again. So here's what this looks like. So there's the shelter, obstacle, and mouse with raw video on the left and a rendering on the right. Sound is triggered and the mouse finds its way to the shelter. Now this is a few minutes later with the same mouse. It's exploring again, and it performs the second escape trial. And again, a few minutes later. Okay, so maybe you could already see this a little bit from the video, but what we found initially was that on the first trial with initial responses, mice perform inefficient escapes where the movement is directed toward the shelter and then they get around the obstacle as it's encountered. Whereas later on in the session, they perform more efficient escapes, which are aimed directly at the edge of the obstacle, which we're calling edge vector escapes. And so a major focus of the study was how does experience allow mice to start taking these more efficient edge vector routes? And what we found by several pieces of evidence was that there's a spatial memory of the edge location that's learned as they become more efficient in their escape paths. And perhaps the most striking evidence of this is the following. So if we allow mice to have experience with this obstacle, the gray tracks are showing their exploration tracks during those 20 minutes. And then we say trigger an escape trial and they'll perform these efficient edge vector paths. And then what we do is remove the obstacle entirely, so it's an open field arena, and trigger more escapes. And much of the time they continue to target this edge location even though it's gone. For comparison, if there's no experience with the obstacle before this, they will head directly to the shelter. And so this spatial memory cannot be explained by visual guidance to the edge, because in this case, the obstacle has been removed entirely and it's an empty arena. In addition, we ruled out another taxon strategy, which is reinforcing turn angles. So we showed that the starting positions and turning angles that mice use during navigation are not stereotyped or consistent, which means that reinforcement of turn angles during navigation cannot explain their edge vector memory. On the other hand, they might be using a cognitive map strategy, which would instead predict that what's important is how much they're observing various features of the environment, such as the presence of the obstacle or the absence of the obstacle after it has been removed. However, we found no relation at all between this kind of exploratory behavior with the spatial memory responses. Instead, we noticed a very particular type of exploratory behavior that turned out to be important, which are escape-like movements that occur during spontaneous exploration. So here's what this looks like for an example. So there's no stimulus here. The mouse is just running to the edge of the obstacle and back to the shelter. And so after observing these movements, we took this raw exploration data and were able to extract those movements specifically, shown here in red. And we found that the occurrence of these movements can almost perfectly predict where subgoals are learned and whether subgoals are learned for escape. So to summarize this strategy, it incorporates elements of the cognitive map. It relies on an abstract sense of place, and learning occurs in an all or none fashion. However, it's also like a taxon strategy in that it's associated with repeated practice movements, and it's relatively inflexible because mice are continuing to target this subgoal even after the environment has been drastically changed. And because this strategy is spontaneously emerging within minutes of entering a novel environment, for the first time, we think it's a fundamental strategy for mouse navigation and could perhaps be used as a stepping stone to more flexible strategies that might be thought of as cognitive map-based entirely. And so with that, I'll open it up to discussion. Thank you.
Thank you, Philip. Now we have some questions for you. Jeremy Forrest is asking, is taxon versus cognitive representation is the same as egocentric versus allocentric representation?
It's overlapping, it's highly overlapping, but not exactly the same. If it's egocentric, then it's a taxon strategy. If it's allocentric, it might still be a taxon strategy because the animal is orienting itself with particular angles with respect to what's in the environment. So you might call, if there's a wall, for example, and the animal is orienting itself at 30 degrees with respect to the wall, that would be considered allocentric, but it would also be considered taxon because it's using a local cue and orienting with respect to that. All cognitive map or locale strategies, as it's called in the O'Keefe book, are allocentric.
Okay, thank you. We have another question for you from Eleanor Duell, asking for, could you please repeat why this strategy is believed to rely on a sense of space?
Yeah, so this comes from, for one, so when we see these repetitive movements, we've ruled out that they're aiming at the visual edge, and we've also ruled out that they're repeating particular movements, so turn angles. But what we did find in analysis I didn't show here is that what best explains the behavior is that they're targeting this particular location regardless of their starting position, even after the obstacle has been removed. So it's very consistent that they're aiming at a particular location, but it's not at all consistent the amount of degrees and angle that they're turning, basically.
I mean, for your the sounds when it is appearing, if you specifically, as far as I see your the final analysis you mentioned, when you are moving it from left corner to right corner, it doesn't change the strategies. They go back. But how many times? I mean, how many stimulations trials they need to develop this map to go back? Is it like 10 trials? Is it quick to have this representation? I mean, how quick they learn it?
So the time scale of learning where most mice learn it is about 20 minutes and 3 trials. So that's what that was the main paradigm we did in our experiment. So 0 trials in 10 minutes. They found the shelter, they've explored a little. Some mice have already, through these spontaneous homing practice movements I've showed you, they've already kind of mapped the subgoal. And some have not. By 20 minutes, most of them have. And in terms of escape trials in our experiments, that's the time of about 3 trials.
If you change this threat area after they learn it, let's say after a few days, to 90 degrees to other corner of the room, do you think I mean, what kind of strategy they are going to do? They are still going to have some, do you see what I mean? If we move this, maybe not 90 degrees, but like 30 degrees, and then see, I mean, I'm just wondering how quick it is to reshape this memory. Is it reshapeable or?
Yeah, so my impression so our work focused on a single session and exploration within that session. My impression is that if you go to a new session, for the most part, they're relearning. If you were to change that within the session, so sometimes we change, for example, the location of the shelter. If you change the location of the shelter, they adapt pretty much immediately, and they're starting from scratch. If you change things in the environment, like removing the obstacle, for example, they basically, how we understand it, our working understanding of it, is that if through exploration they manage to practice a correct route, then they will update their essentially their map of subgoals. If, however, through exploration they haven't done that, they haven't explored in the right way, then they'll continue using the old strategy, basically.
Okay, thank you. We have more questions for you. Raunak Basu is asking, but can these be explained by stereotype action strategies? In spite for not repeating single angle of turns, etc., they can be higher order action strategies in premotor cortex. Premotor cortex.
So I guess it depends on what he's meaning by stereotype action. So if it's not, the way we're thinking of it, the mouse is starting in a certain position, and the way the behavior is triggered, we trigger the stimulus, they turn toward their target location and run toward it. So they can either be memorizing, this is basically egocentric versus allocentric. If they're memorizing the egocentric action, that would be then stereotyped or predictable in some sense. If they're memorizing an allocentric target, then we wouldn't be able to predict their egocentric action, but just predict what they target. So I'm not sure if that answers the question or not, because I already said that. So if it doesn't, feel free to clarify.
Yeah, thank you. And Hunglo says, great talk, and asking for which brain region will be most likely involved in this edge vector escaping, pure PAG, or will this be the crosstalk between PAG and spatial memory related brain regions? What do you speculate?
So, I mean, work from another preprint from our lab is already implicating retrosplenial cortex in directing the angle of the mouse to the shelter and superior collicus as well, and the connection between the two. So even in the more elementary task of orienting toward the shelter, where it's a single step route, already cortex, association cortex appears to be involved, and probably forebrain and hippocampus will be involved as well, because these are allocentric representations being used. So I mean, I expect the whole suite of navigation related areas that have been found in previous work with foraging will be engaged as well.
Okay, thank you. And the final questions for you from Eleanor Duval. Have you shown that this behavior depends on distal cues? For example, rotate a salient distal cues between outbound and inbound paths?
No, that's a good question. We haven't done that experiment. My guess is that it does not. And the experiment that we're doing as kind of a proxy for that, or not a proxy, but it's a related question, is if we turn off the lights altogether and remove all visual cues, are the mice still able to perform this task? And we found in certain cases, after they have experience and have learned the subgoal, if we're turning off the lights and there are no more visual cues, they're still able to perform the task. So I'm sure that distal cues will be incorporated if they're available and they're salient, but I do not think they're necessary for this behavior to occur.
Okay, thank you very much.
Thank
And
you.
I hope we